depending on your location. I want to welcome you to today's webinar on soil data aggregation in R. And today we have three presenters, Dylan Bodette, Stephen Roker, and Jay Scovlin, all soil scientists within the agency in various places, California, Indiana, and Montana. Today's webinar is in Adobe Connect, and you can enlarge the display using the icon with the 4 arrows. And to shrink it back down on your machine, you push your cursor to the top of the monitor, and you can shrink it back down pressing that same icon. I do have a chat window open. If you have some general comments you want to make back and forth, you can utilize the chat. Otherwise, if you're going to submit a question for one of the presenters to answer, I'd invite you to put that in the Q&A pod at the lower left corner of your Adobe Connect window, and then we'll take those questions as they come in the end, unless one of our presenters notice there's a good topical question and they can pick that up and answer it on the fly. Today's meeting is being recorded and will be available within a week on our National Soil Survey Center YouTube channel. So it'll be publicly available. If you want to do a search in YouTube, you'll be able to find it very quickly. Just wanted to mention that SSSA had a workshop on using R, and it was almost $200. So good value on today's presentation. Everybody that's an NRCS user has access to R. If you want R, you can get it installed on your machine. And with that, I'm going to turn it over to our presenter panel to start our presentation. Dylan.
All right. Well, I'll start the show. This is Stephen here. For those of you who that title is not clear enough, hopefully that'll become apparent in a minute, but since a lot of, because there hasn't been a webinar that I'm familiar with thus far that has really introduced R, we'll be doing a little bit of intro to R and intro to the data aggregation process that we're outlining here. As Sean said just a minute ago, come on, switch screens. Every NRCS user has R. It comes installed on every USDA machine with a NRCS install also. And if you were to open up, your desktop and go to your start programs, you'd see, 2 R programs that you can use. The one that I have open here is referred to as RStudio. Typically a little bit more user-friendly, so that is generally what we would recommend to most users because it has this sort of multi-window out panel display going on here. So, what is R? For starters, it's generally thought of as a statistical program that, you can do your typical means and standard deviations and your linear models and all that. The closest analogy sort of in the commercial market would be something like S+ or SAS. R is actually a derivative of S+ that broke off many years ago when AT&T wanted to give away the license, essentially. And since then, it's kind of morphed into this basically super program that has all this added functionality that will, some of which we'll address here in a minute. So, it's a calculator. If you open it up and you type in the R console, you can, do 1 plus 2. It's also got a surprising number of GIS functions in it. It's almost a GIS all by itself these days with a lot of the extra packages or extensions that you can load in it. Another way to think about it, if we sort of stack R up against NRCS, against ArcGIS, each of these different programs has sort of their inherent strengths, and they all do different things, but the great thing about them is with the connectivity between all the different softwares these days, we're actually able to leverage all of them within R. So I can call another GIS from R, I can call NRCS from R, and it's not important that, for example, that you're able to do all of your analysis in ArcGIS or all of your analysis in NASIS, because each of them has their inherent strengths. What we'd really advocate is sort of, using sort of the three in combination and getting the best out of each of the different systems. What can R do? When I first started off using R, not quite 10 years ago, that first bullet there of base R functions is essentially, what was available for what was available out there. And as I said, your basic summaries, your basic statistical models. There was some GIS functionality even back then. But since then, thanks to the fact that R is an open source program and it has tons of user contributed packages, some of which myself, Jay, and Dylan are contribute to the R CRAN library. There's a host of them out there now that are specifically designed for soils. Like you can plot a soil texture triangle, you can plot soil profiles. Digital soil mapping is a big one these days that seems to be everyone's jumped on the bandwagon. It seems like when I started again, like almost 10 years ago, nobody was using it. Relatively few people, I should say, were using it for, say, digital soil mapping. But now it seems like it's kind of in vogue, and everybody's doing it. Some of the things we're going to touch on here in a minute is one of these. This is essentially a profile plot with the probabilities of different horizons broken out. And one of the neat things that Dylan's come up with is a way to sort of estimate, based on some of the things we're going to talk about here in a minute, what would be the most probable horizon for a given depth. And then, potentially using that probability down the road. If we can segment out the different horizons and sort of group them all, like all the A horizons, be that an A1, an A2, an A3, we're able, Jay's going to touch on this again in just a second, but we're able to estimate sort of useful ranges and ranging characteristics from the different soil properties that we collect. All of this data here coming from NASIS. Before any user can take advantage of a lot of this functionality, though, there are some, what do you want to call it, growing pains or whatever. The first time someone sets up, they'll have to install these packages: AQP, RODBC, SoilDB. And the reason they have to do this is because they're not base R functions. So they're user-contributed set of functions that add specific functionality outside. They live inside the R ecosystem, which is referred to as CRAN, so they can be installed straight from the program. But anytime you open up R Studio or R for the first time, you'll have to load these and then update them as you see fit. Updates come out relatively frequently, depending on the package. But every time you load R, you'll also have to load the different packages because they're not automatically loaded unless you override that default. Another thing for users to read soil data straight from NASIS out of their local data set into R, they need to set up an ODBC connection. There's a bunch of job aids, some of which are hyperlinked right here in this presentation, where you go into your computer system settings and set up a connection. There's multiple job aids out there for this. There's a very good one on the the NRCS US Soils Job Aids page, so we're not going to go into that now, but after you would have set up an ODBC connection, all you would have to do is enter these commands such as we have here, paste it in the presentation, run a fetch command, and it'll automatically detect your ODBC connection, and you're able to read data straight out of NASS into R. You can also read component data, and there's a host of functions as well. So with that, I'll turn it over to Jay.
Thanks, Stephen. Hey, this is Jay, and I just wanted to mention one more thing about the connection to NASS through the Open Database Connection. And that connection pulls, using the SoilDB package, it pulls data from a NASS selected set, and so you have to go in and manage your selected set accordingly, and make sure that you have the data that you want loaded. And then it's a really convenient way to shuffle that data, basically pull that data directly into R. And in the process, it has some kind of cleaning methods, and it sort of makes that data a lot easier to work with. So yeah, that's kind of that connection with NASS and pulling data from NASS is kind of at the heart of this whole process. So I'd like to talk a little bit about the aggregation of pedon data and some of the common problems and challenges that we've encountered in this process, and just background on it. As you can imagine, there's many different description styles out there. I swear it's probably true that no two soil scientists would go in and describe the same pit exactly the same, right? And so we have the two camps, we have the splitters and we have the lumpers. I was joking the other day that the only reason that Dylan, Stephen, and I can still work together with each other is that we've never actually been in a pit and described the soil together. So, you know, things get a little intense when you get soil scientists in the pits. But we have all kinds of description styles, and so we have also, in that, we have different uses of transitional horizons. Some people like to pull them out, some people don't. They may, you know, one person might pull out a BC horizon, another might pull out a CB. So you have all these different methods just in the process of describing the morphology that exists in the soil. And on top of that, we have legacy methods or legacy nomenclature. And so if you're dealing with soils data that's, you know, say before the 80s or early 80s, you might be looking at some old horizon nomenclature, which is considerably different from what we use now. And so you have to meld that, and you have to mesh that in with our more current methods on that. So there's issues with that. And then the other thing, the last thing is the, you know, in the case of this diagram that we have here, the black labels on the side of the profiles are what was actually described, and then our middle layers would be these kind of generic generalized horizon labels that we would correlate those horizons to. And in this case, you know, we have a soil that's over a bedrock or a paralithic contact and a lithic. And so, you know, we're describing that soil down to that contact. But in a lot of cases, you have, you know, very deep soils, and those soils maybe have been described to varying depths, and maybe not all the way down to the full 150 centimeters. And so you have just different obstacles and challenges in dealing with this data, because if you're trying to compare lower horizons in the profile, you might be having troubles because the data gets a little thin down there, past 100 or past wherever it was dug to. So those are some of the issues that often come up in this kind of perennial issues, and this process seems to address a lot of those. Next slide. Thanks. So the concept here is to use these generalized horizon labels, and essentially what you're doing is you're running a process of microcorrelation across horizons of a set of profiles. And so you have this collection of profiles. One of the first things you need to do is determine, okay, what is the core concept that I'm trying to convey with these generalized labels? And how do I want to deal with some of these different horizonation issues that occur, such as the use of transitions or different older nomenclature that you've got to deal with. So how are you going to correlate some of these? And you can see in this diagram, I'm not sure if you can see it or not, but we've got a BA horizon in the generalized scheme of things, and then we've got an AB that we described in one of the profiles. And so, we're going to correlate that AB to a BA because that's more dominantly what we have in the soil concept, and that's what we want to convey. Could go the other way as well. It just depends on what you're trying to convey. Another issue is redundancy in horizonation. So we have potentially 3 BTs here. Is that really necessary in the core concept that we want to convey? And then the other thing is, occasionally we'll have in a profile, we'll have outlier horizons, or say a stratified horizon, something that's just sort of oddball in there. And so, what do we want to do with those horizons? And we have the possibility here to either to correlate them in this process and apply a generalized label to them, or we can just basically let them fall through the analysis and fall out of the analysis if we think they really don't fit the concept. And that applies also to certain soils. I mean, say we have soil in this group that had been identified in this taxa or this component name, and it just didn't fit the group as a whole. We can drop that whole profile from the analysis. But we're trying to keep as much as we can and to correlate across these horizons as cleanly as we can. Next slide. So as far as a workflow, most of these steps have been done to create the reports that we're going to show later on in the presentation. And so this process is kind of at the core of how we create these data summaries. So our first step in this is to select a set of horizon labels, generalized horizon labels that best represents the group, the whole group, and the concept, the morphology concept that we want to convey. Second would be to assign the labels, the GHLs, to each horizon, looking at whatever data we need to look at and comparing that data across those generalized horizon labels. So, and then we're evaluating these assignments, and we can tweak them manually. One thing I want to stress in this process is that there's a lot of user control. So it's not a push button type of thing. There has to be a lot of operator control in this process. It's just the way it is. And then we're keeping track of those final assignments of the generalized horizon labels first in a text file, but that text file is able to be uploaded into NRCS, and that goes into a field in NRCS called the component layer ID, comp layer ID field in the pedon data structure. And so we're able to actually make these GHL assignments and then trap them in NRCS, which they can then be used in NRCS type reports, or just at least stored in the so that knowledge is stored in the corporate database. And then we can go beyond step 4, and we can use these GHL assignments, the horizon assignments, to estimate a maximum likely depth for horizons. So this is sort of an idealized profile based on our collection of profiles. And we can, across those depths, we can compute ranges of characteristics for different soil properties. We have a link down here at the bottom to an extended tutorial on that. Next slide. So in this process, we're making some assumptions, and they're fairly straight ahead assumptions. So we're saying that a collection of pedons for a component should represent the aggregated data, and this is sort of looking at things from a data-driven, more data-driven type approach. There's going to be some kind of generalized horizon process required in order to make these summaries possible and to make the aggregation of the horizon data in these collections possible, and then to output that to reports. The next thing is the low, high, and RV values need to be, or they should be tied to some metric that's sort of a standardized metric, and so we've kind of looked at the 50th and the 95th percentile as being an option of that. And then linked pedons represent examples of the aggregated data, but the aggregated data should not be limited to just a very select set of linked pedons in our view. And so it's important to stress that these reports and these data summaries are not, we're not generating component data here, straight out of the box. We're not push button, streamlining anything like that. This process requires a lot of operator control, and it's really just a starting point for looking at the aggregation process to component data. I think I'm going to turn it over to Dylan here, and he's.
You guys hear me?
Yep, it's good.
Okay, can you hear me, Sean?
Yes.
Okay, great. Hi, everybody. This is Dylan. So I'm going to backtrack just a little bit, and we'll go over a little bit of some of the terminology and figures that we might be, that we will be using the next couple of slides. As Jay mentioned, that we are proposing, at least for now until we come up with a better idea, the use of the 5th, 50th, and 95th percentiles for the so-called low RV and high values used to populate either component data or to come up with range and characteristic summaries. And very quickly, in this figure here, there is a frequency distribution along the bottom, the black line of a normal distribution. Right above it, there is a blue box and whisker plot of that same distribution. And the box and whisker plot is a very convenient way to show ranges in data. These little blips here up on the top, these are the values of, say, a hypothetical range in sand content, say, collected from a bunch of different samples. Within the box and whisker plot, this middle bar here represents the 50th percentile, which is also called the median. The edges, or the kind of the main body of the box, represent the 25th and 75th percentiles. Within that box, that range is commonly referred to as the inner quartile range, because this 25th is the first quartile, 75th is the third quartile of the data. And then these whiskers extend out to, on the low end, the 25th percentile minus 1.5 times this inner quartile range, which represents, depending on the distribution of the data, typically somewhere between the first and 5th percentiles. When we talk about percentiles, it means that, say here at the 25th percentile, it means 25% of the observations are lower than that value. For the median or the 50th percentile, 50% of the observations are below that value. Another way to think about these things is 50% of the time the data are going to be greater than that value, 50% of the time the data are going to be less than that value. So the 50th percentile represents a central tendency. and for roughly normal distributions approximates or is close to the mean, which is this central green square here. The green squares at the edges here represent the mean, well, 2 standard deviations subtracted from the mean on the low end and 2 standard deviations added to the mean, which is a typical way of expressing spread around a central tendency. And for roughly normal distributions, that's a pretty efficient way to represent central tendency and spread, the mean plus or minus 2 standard deviations. Unfortunately, as distributions potentially shift away from normality, those are no longer efficient or even realistic estimators of central tendency and spread. In this case, we have a long-tailed distribution. This looks like say a distribution of soil carbon, where you have a number of very large values and possibly an order of magnitude more values clustered around 0. In this case, the central tendency here, the 50th percentile or the median, is quite a bit different than the mean, and the description of spread around that tendency is adjusted to accommodate this long-tailed distribution. What that means is that there's the lack of symmetry around our central tendency is automatically built into our proposed low, RV, and high values. So the 5th percentile stacks up pretty close to 0. The 50th right here, somewhere between 0 and 1, and then the 95th percentile better accommodates that small set of values that are a little bit closer to 7. So the mean plus or minus 2 standard deviations on one hand gets close to describing this new interval, but on the other side, clearly 2 standard deviations subtracted from the mean gives you negative values, which doesn't really make a lot of sense here. So getting back to our proposed use of the 5th, 50th, and 95th percentiles for the low RV and high values, essentially there's no assumptions of a distribution, and they have a very simple probabilistic interpretation. 95% of the time, the data are going to be less than the 95th percentile. That's a pretty clean and easy way to describe to non-specialists what that means. So within the next couple of slides, we're going to use a sample data set that helps standardize some of the descriptions, but clearly all of the additional material we present can be applied directly to data in your selected set in NASIS. So this sample represents 15 pedons correlated to the Loafer Creek series. It's fine loamy mixed superactive thermic multicapital almost exactly the same taxonomy as 90% of the other soils you find in MLR 18. This one just happens to have a lot of data, and so it was convenient to use. And the sample data set is included with the Soil DB package. So you can call this data set up at any time to test reports that you're writing or to tinker around with. As a side note, one of the convenient aspects of our packages is that they contain functions, or you can think of them as verbs for doing things. They come with sample data sets that allow you to practice with those functions. And they also come with manual pages written for the functions and for the various sample data sets. And they're full of examples and demonstrations. So one of the best ways to get familiar with R is to tinker with some of the sample data sets. So within the context of this generalized horizon label framework for aggregating soil profile data, probably the best thing to do is consult the official series description for some initial ideas. Checking to see if there are existing range and characteristics that can help guide the assignment of these labels. And sometimes the horizon structure from the series description is a good starting point for this generalized horizon label template. So within our sample data set, again, Loafer Creek, the first thing I usually start with after looking at the OSD is just tabulating horizon designations by frequency. I can see, well, I've got a lot of A's, a bunch of BT's, and then subsequently fewer and fewer kind of stranger and stranger mixtures of horizon concepts. We've got discontinuities, a couple discontinuities. We've got a couple transitional horizons here, a BA and a CBT. We've got a BW. Okay, well this gives me an idea of where most of my data exists, and then over here to the right-hand side gives me an idea of those horizons that are likely going to need to be correlated to some of the more common horizons, or possibly kicked out if they're oddballs. Sometimes sorting them alphabetically is helpful to see what's going on. Another way of viewing these data using the box and whisker plots that we just talked about is to generate a box and whisker plot representing the ranges in horizon midpoint depth for every one of the possible horizon designations. So we can see here, well, it looks like that set of CBT horizons overlap in depth approximately with these BT3 horizons. That gives me an idea that these two may be candidates for correlating together if I have additional information to support that. Same with this 2CR and CR. Well, maybe I should investigate that lithologic discontinuity. Is that real? What's that all about? Same with the various transitional horizons here. So figures like this can be generated with just a couple commands in R. We're not going to show the syntax right now, but there are links within this presentation to an expanded tutorial on this topic that includes the narrative that we're giving now, along with the code to implement the ideas, and then the resulting figures, all based on that sample data set. Another way to kind of look for horizons that could be correlated, we can plot the range and clay content according to each one of these horizon designations. Checking to see here, well, let's see, this BT2 and our BT3, those have very different clay contents. Well, maybe those shouldn't be correlated. Rock fragments is another property to look at, but this can be done for any horizon level attribute that exists in NASAS that you might find important. pH, the quantity of redox features, you name it. Okay, so once we've reviewed our data, our OSD, and any accessory information, it's time to come up with a set of rules using something called regular expression that we can associate a series of existing horizon designations with one of our new generalized horizon labels. And these regular expression patterns, they look a little bit complicated, but with a little bit of practice, it's a very powerful way to match a whole suite of horizon designations and assign them to a single generalized horizon label. So in this case, we've got, let's start with this BT here. So we're saying anything that contains BT1 anchored to the right-hand side. So this would match just about any permutation of BT1. BT2 anchored to the left-hand side and the right-hand side would then filter out any lithologic discontinuity. So by anchoring this string to the left-hand side, we no longer match numbers in front of that B. For the BT3 horizon, we're going to pick up basically we're picking up all the kind of weirdos that occur at the bottom of the. Again, specifying these patterns takes a little bit of practice, but we've thrown up a bunch of examples, and this is kind of an interactive process. The vertical bar here means or, so we're saying matching this pattern, or this pattern, or this pattern, and so on. The next step then is to compute a cross-tabulation of the original horizon names, which should be the column headings here. Compare those with the assignment of our generalized horizon labels, which would be the rows in this case. So if we look here, we see, well, there's a total of 15 horizons correlated to an A, and of those 15, all 15 are matching just a simple A horizon. Well, that's a pretty easy interpretation. Same with the BT1 and BT2. I guess there was quite a bit of consistency within these pedons. For the BT3, we can see that there's 2 BCT that's been correlated to this, and a number of other horizons. This is kind of our bottom of the profile garbage can, or catch-all horizon. Whether or not that makes sense in every context is totally dependent on the data, your understanding of the concept that you're trying to convey, and hopefully looking at things like the series description and whatnot. So this cross-tabulation is one of the simplest ways to check your assignment of generalized horizon labels. This is the template over which we're going to aggregate data to cross-check that with the original horizon designations. And then it's useful to check in on this not used row, which represents those horizons that haven't been caught by any of our patterns. In this case, we didn't specifically deal with any of the BA horizons, and we didn't deal with the BW horizons. For this concept, they just weren't quite useful enough. So we didn't provide a rule. They fall through the cracks. It's convenient to plot these things graphically. Again, it's just a couple of commands in R. We won't go through the syntax here, but it's on the expanded tutorial online. If we color the horizons, the original horizons, using the new generalized horizon labels, we can see some patterns that typically those lower BT horizons and associated BCT type transitions are all falling approximately within the same context of the profile, but they do occur at different depths. And then those horizons that are yellow, these are horizons that don't have a rule, a pattern that matches them to a generalized horizon label. So these horizons will not be included in any kind of summary at this point. So we have to think about, what do we do with those things? Should we specify a new rule to pick them up? Are they important, or should they be correlated to something else? Once we're happy with those, it's useful to take a look at the range in depth that the various generalized horizons span. Clearly, there's going to be overlap because we know there's variability out there. Any given series or component concept is going to have a range in depths or thicknesses of the various horizons. Overlap is to be expected. Excessive overlap might indicate that we've done a little bit too much correlating. We've included too many different horizon designations into a generalized horizon label. So this is a pretty reasonable amount of overlap, I would say. And we'll just briefly look at this diagram here. The expanded tutorial goes into this in quite a bit of detail. In some cases, you may want to evaluate the assignment of your generalized horizon labels over more than just a couple variables independently. It could be that, say, the depth of the horizon, the clay content, the rock fragment volume, the pH, and maybe the color all contribute to your decisions on how the original designations are correlated to your set of generalized horizon labels. And this right here is something called a principal coordinates plot, and it's a dimensionality reduction technique so that you can take depth, clay content, rock fragment volume, pH, what have you, and compress that down to a two-dimensional figure where you can start to look for, you can look at the proximity of these different points. Each symbol is a horizon. It's labeled with the original horizon designation and colored according to the generalized horizon label. Again, some amount of overlap is to be expected, but there are cases where perhaps too much overlap suggests that there's a correlation issue. For example, right here we see a BT2 and a BT1 generalized horizon. They're sitting next to each other here, but they've got different labels. This green one here should probably be correlated according to its soil properties to that BT1 generalized horizon label. Underneath, the pedon IDs are printed so that you can go back and check the actual data. Once you're happy with your assignments and you've evaluated the data, it's a pretty simple matter to run a couple of commands in R, which will save your assignments out to a text file. There's a calculation analysis under the pedon horizon folder called Update Horizon Group Aggregations using a text file. It'll look for that file specifically here. You check out your pedons, get them in the selected set, or I guess what, set up your selected set, check them out, run the calculation, and all of your hard work will then be saved back into NASAS. If there are some special cases or outlier horizons that you need to manually adjust, then you can adjust those as needed in NASAS for a permanent record. And again, there's a link here to that full tutorial that goes over this in quite a bit of detail. So I'll turn it over to Stephen now.
Okay.
Let's see, I think you may need to share your screen.
You got something?
There you go, it's all yours.
All right, well, you know, the whole generalized horizon labeling scheme, so what? This is kind of what I have in front of you here is kind of the so what, like, you know, why would we want to go through all the effort to draw those?
Stephen, you're not sharing anything right now.
No, why not?
Oh, here we go.
Yeah? Okay. Why would you want to go through the effort to draw all those microcorrelations? Well, from the standpoint that I think Jay touched on earlier, and I showed some of those early figures, was if you can draw all those sort of microcorrelations, as he called them, then you can, you know, in NASAS or in R here, you know, aggregate using your horizons and then do sort of a data-driven summary of everything that is associated with an A horizon. You can easily compute some kind of summary, be it a mean or a median of, you know, what's the average rock content, what's the range in average rock content, what's the range in rock content to any of your other horizons. And one of the functions I talked about earlier in R, the reporting capability, is that we're able to sort of write these HTML stylized reports where they're, you know, once the generalized horizon labels get taken care of, it's really just a push of a button, such as you would do in NASAS for any of the reports in NASAS, although these ones run in R, and you can easily compute a summary, you know, using all the extra functionality there to compile it all into one document. So you could have, you know, I could have called this an OSD report because, you know, I don't know if it's showing my mouse. Oh yeah, it is. This is looking at a soil series that I was working on last year in California. So I'll scroll down through this. As I've set it up to begin with, you know, it pulls in a little map with the coordinates sort of plotting everything out. Right now it's a pretty basic map. If it, you know, overlapped a couple counties, it would show the county boundaries, but for right now it's not anything fancy. If we were to keep going down, it would have some more of those sort of profile plots, and as Dylan discussed, you know, the coloring matching the you know, given generalized horizon matches that we put together. It would give a, you know, short synopsis of who described them. You know, in these instances, it was myself, Leon Lotto, and Carrie Ann Howdyshell. So we get to see the classification, the particle size, the user pedon ID. So this is just sort of a, you know, quick synopsis of what's in our selected set. So this is everything that's getting, you know, run through the report and, you know, aggregated. We get to see what was the range in those pedon's elevations as they were observed in the field. Here's a 5-number summary, which I preferred. It has the minimum, the 25th, the median, the 75th, and the max, in addition to the total number of pedons here. So this was a total of 11 pedons here. Parent material versus landform, slope shape, so, you know, it's on a hill, shocker, it's linear on average. We're out in the desert, go figure, everything's well drained on average. A summary of the, you know, rock fragments on the horizon, or on the top of the surface. And then we get into some graphics, breaking out what's reflected in the table up here. And now we're getting back down into the sort of stuff we were covering just a minute ago with the generalized horizon labels. Again, this being the column headers being the original horizon descriptions, and then what they match up with in our generalized horizon scheme. So I chose A, ABK, and etc. And scrolling down some more, now we get to see some of the good stuff in terms of what's the average clay content of an A horizon. Well, in this case it looks to be about 8, but it can range anywhere from 6 to 20. 20 being probably on the high end and unrepresentative for the series. But if we throw in the sort of similar soils or well, the similar soils, you get to see what the full range of anything that's correlated to this series looks like. There's also pH and color, in addition to rock fragments. And then these numerical summaries are great, but it's even better to get to see how these things look in graphics. So the range becomes a little bit more intuitive right away when you see here's our clay content, and there's a wee bit of a bump with the BA, but it quickly decreases as we go down. Same thing with the gravel too. There's a heck of a lot on the top, on the surface, quite a bit in the A, but then it jumps up again down in the BK. So all these patterns kind of, one at a time jump out. And then we can also see, what's the average soil texture for the A horizon. In this case, it's pretty clean cut. It looks like it's, pretty much sandy loam. Sandy loam. Once we get deeper, it gets a little bit, wider. We go all the way to a loamy sand, to a sand, to sandy loam, and etc. This one down here just has the modifiers added on as well, so we get to see, the different combinations of cemented and etc. Dry hues, moist hues, effervescence. This is a calcareous soil, so it's not shocking that there's a lot of violent effervescence going on all the way throughout the profile, but in some cases it's only strong or rarely very slight. So again, by having all this data together in NRCS, you're able to not just look at 5 pedons, you could look at 100 pedons, or however many. Obviously 100 would be a little bit excessive, but I think you get my point. Depths of the horizons as you might populate them in NRCS, the highs and lows for each horizon. Its thickness, we get to see that graphically again. How does that plot out? Like, obviously the A horizons are really thin, but the BK can actually overlap the BKK in some instances. We can see the range of the diagnostic horizons as they're populated in NRCS. So, particle size control section always starts at 25 and, generally ends at 100. For this soil, that's not shocking. Range of the ochre pedon, more graphics. That one's just looking at pedon data. If I want to go in and look at lab data, which is, obviously more valuable. This is for a soil series out here in Indiana. And, now this map gets to be a little bit more useful because, we're seeing its geographic spread, the points here, over, a series of 3 states. And for some reason, there's one little outlier up here in Minnesota. I don't know how that one got up there. That could be either, the wrong coordinates, or, for some reason, someone thinks they found it in Minnesota. But, the other point of, a lot of this stuff being, this is a good QA tool too. Like, I'm sure people have seen, maps like this before where, you plot your pedons and they end up somewhere in the ocean or the Arabian Peninsula, you name it. So if you use this as a QC tool, you can catch errors in your NASIS data very quickly. This one doesn't have it, but one of my favorites, one of these graphs here, the depth of the horizons goes all the way down to like 8,000 centimeters or something like that, which is an obvious error. Again, a summary of, the data in our selected set. For this one, it looks like we had a total of 44 laboratory pedons for this one series, which actually isn't a very common series, so that's surprising that there's that much laboratory data associated with it. The cross comparisons again. The output of this one is a little bit funny because it's formatted as a text as opposed to straight HTML. But I'll just scroll down to the graphics again. This is giving me a summary of all the laboratory data for this one series right here in this this data set. The only other way we can go about summarizing all this data at once would be to slice it by 1 centimeter intervals, which would also generate a similar summary, but would you know at any given depth, well, what horizon is that coming from, obviously. So again, if you go through the process to put together sort of this matrix of generalized horizon labels versus the horizon designations in NASIS, you can put together some really quick summaries of what's in NASIS. So all the more reason to enter pedons into NASIS if they're of high quality and representative of your given soil. Another one I'll just show real quickly, and then I'll turn it over to Dylan. Is these reports are just pulling data out of NASIS, but we could also look at map units as well. So we could run these same kind of summaries on sort of an SDGR project, and this is an SDGR project down here on the, well, this is Kentucky, this is Indiana, and this is Ohio, and see where is it mapped, and what do we know about those map units? All using, again, sort of similar metrics to evaluate all of these map units. So I think that should cover it. I'll turn it back over to Dylan.
You got ready for some questions?
Yeah, that's probably the best thing, is to answer some questions. And just before we do that, I just mention again that we'll make sure that, this presentation has a number of links to outside data sources that go over what we're talking about in gory detail, but we'll make sure that those links get posted.
All right, the first question I see, do you also consider the geographic location of the pedons or component estimated profile in question during the correlation process, the lat to long?
Well, the great thing about this process is it's totally extensible. So we've presented kind of a framework, and then some of the output reports that are totally customizable. But sure, you could incorporate any and all variables you think are important to that series or component concept.
And there's a question here from Edwin Muniz. With the rejects utilizing the caret symbol in the code, how will that work with the human transported material horizon modifier? We use the caret symbol for an actual horizonation label too.
Yeah, that's a good question. So in regular expression, you have what are control characters, or these special entities, which would be like the caret and the dollar sign and the percent sign. And if you would like to use one of those as the actual literal caret or literal dollar sign or what have you, you can escape these things with a backslash. And that is a great idea for maybe a very short tutorial on the use of regular expression to match horizon names to accommodate some of those special characters like the prime or the caret symbol. That's a great question and reminder. Thank you.
And the next question, do we have these methods documented in National Soil Survey Handbook or cited in our Sergo metadata yet? And I know you mentioned job aids available. Do you want to expand on that?
If you guys don't mind here, I'll share my screen and then you jump in as necessary. These things there's quite a bit of documentation on the project website, this AQP project website. Lots of tutorials. Many of the job aids link back to this page. That's a good place for documentation. There are a couple of papers written on this topic. Those are good references that can be cited. And I believe that a lot of this material will end up in the Soil Survey Handbook in a chapter that's currently being written. But you guys chime in if you've got some more.
No, I mean, there are, some job aids on the USA Jobs website, but, they're only as, some of them are written by us, and I should say all the ones on this topic are written by us. I have some custom ones written just that are shared on my region's SharePoint, but they haven't made it on the USA Jobs site yet. Some of them are in that last link up on the screen here now. That's a GitHub repository. But
yeah.
They don't kind of exist in the current, within the current NSSH handbook and all that to date. So I guess we're proposing that they be incorporated.
Right. Most of the documentation has been collected here, with some of it percolating up to the job aids page. But yeah,
I think another comment is that the process of constructing a lot of this material for R, the documentation is kind of baked into the packages as well. So, just going into the packages and going onto the package website, as Dylan was mentioning, that's a great way to start.
Another question we have online is, has there been any talk about having an R training session geared specifically to soils analysis? And I know that we've got the online resources that you pointed out here for some of the classroom courses. We've attempted to incorporate some exercises using R, but have you guys talked about any other sources for R training? I know SSSA was putting on an R workshop. Of course, that was almost $200 to take that.
Well, right now, Tom DeVello is kind of trying to build momentum for just that sort of training course that you're talking about now, because there are so many topics where this would fit into within say the soil correlation course or the basic soil survey, or any number of courses. But yeah, it just statistics is usually the last thing that most people want to think about, so it's always kind of swept under the rug in the past, I guess.
a lot of it has to do with interest. I think the three of us and then a number of others probably listening right now would probably be quite interested in putting that kind of training together, but it all comes down to interest. Just about any one of the tutorials on that AQP website could be turned into a full-length webinar. There's a ton of documentation that's been written with examples applied to analysis of KSSL data to SDJR stuff.
I think we have time for one final question, and it relates to ESD development. So how can AQP and other tools be applied to ESD development? Component and or map unit level data, any linkages to ESIS or MS Access databases? In other words, not NASA's.
Well,
ESIS, ESIS is a hard one, but Jay, I think you could speak to the access thing.
Yeah, the SoilDB package allows access to access databases and to pedon databases specifically. And so, but I mean, I think that there's a lot of application here with just pulling component level data, summarizing either component level aggregated data over ecological site concepts, or looking at it from the lowest level in the data, the pedon data and the corresponding veg data, and then aggregating that data up in these reports to describe the soil properties for specific ecological sites, and also even for their reference states and different states that they may have. So I see a lot of area to work in that arena with this kind of material.
I'll throw in one final thought related to that, and that is a lot of folks may be asking, why do I want to learn another language, another environment? And Jay and Stephen and I have talked about this a lot, and that R is a very convenient integrator of all the different tools we already know how to use. And learning the language is a convenient vocabulary for describing just about any possible analysis, summary, modeling of our soil and ecological data. So it's more than just a fun, exciting, shiny new thing. It's an actual vocabulary for describing repeatable, high-quality work.
Well, I want to thank our presenters today, Dylan, Stephen, Jay. Very good presentation, and used an example I think anybody with some field experience can relate to with the various horizon designations. So great job, guys. And I'll have the webinar posted within the coming week to YouTube. Thanks, everybody.
Thanks, Sean.